Assalamu alaikum. Welcome to this live stream. We are doing part 2 of this new series on orphan genes. Over to you, Paul.
Okay, I need to share,
Sure.
and I'm not sure which, they've changed the software, so I'm not sure which button that is.
It's the plus with
Oh, I
the
see. Share screen. I got it.
yeah. Should be coming up on my end.
All right. It doesn't show. Oh, here we are. Got it.
There we go.
And my title slide. Okay. So this was originally a 3-part series, but I didn't finish.
Sorry, your slide's not coming up. Something else is coming up.
What do you?
You've shared your screen instead.
Oh, shoot.
No worries.
All right. Hang on just a second. Everything happens at once. My cat needed to go out.
No
All right.
worries.
All right. So, I'll start again. Present slides. Your computer. No, I don't want to upload a file. I'm very sorry. This.
No worries. No worries. It happens. While you get that ready, what I could do is I could just jump off and start posting if you need a few minutes.
Well, I'm trying to figure out, can you see my title slide now?
No, I can only see the screen.
I'm not sure what to do.
No worries. Take your time. I'll just be posting this stream so more people can join.
All right. Okay. Well, I could try upload file.
That could work.
It says it's processing the file right now.
No worries.
All right.
There we go. Perfect.
Okay. Are we live?
Yeah, we are. We are
Okay.
live.
So this was supposed to be a 4-part series. Excuse me, a 3-part series. It's now a 4-part series because last time I had too many slides. I've shortened the presentation, so we should be done comfortably within 45 minutes.
Right.
And it will not run over. But I encourage the viewer, if they're curious about what happened in the first part, to go to the YouTube link there. And I will just briefly summarize the main points of part 1. The first is that within evolutionary theory, the distinction between homology, that is similarity due to common ancestry, and homoplasy, which is similarity not due to common ancestry, may be unstable. That distinction may not work especially as we go back in the history of life, because of the deepest theoretical commitments of what I'm calling the naturalistic program of explanation. And that is the attempt to understand the living world without respect to design, teleology, plan, foresight, any of these notions of purpose that I think biology needs, but that were, according to the standard history, driven out of biology in the mid-19th century by Darwin. So the naturalistic program of explanation has what I will characterize as conflicting commitments at a deep level that have as a consequence unstable concepts like homology. And that's problematic because the science ends up asserting contradictions. Second major point, evolutionary theory is not mainly a theory of similarity. Similarity was known long before Darwin. It was known and observed by Aristotle, not in an evolutionary context. Similarity is mainly a theory of transformation over time. And because of that, evolution needs to be able to document or understand the pathways of transformation that are its major empirical claim, that species are transformed over time, that they split into two daughter species at speciation events. There's transformations that occur in that process, that the chordate body plan, let's say, from the original chordate to the, tens of thousands of different chordates that we observe today, that there have been transformations all along those lineages. That needs to be documented. The problem is, and we'll look just at proteins today, describing the starting set of entities that are going to undergo transformation runs into a problem that when we come to biological reality, for instance, what is necessary for the cellular state, we may find that it is impossible to describe our starting set of entities which are going to undergo transformation. We arrive at what I'm calling basic functional thresholds beyond which we cannot see. And this ends up being a problem for evolutionary conceptions of homology. So those were the main points of part one, and I'm going to elaborate on the third point today. Okay, so what happens as you go back in time for any group of organisms, let's say for the mammals, or for vertebrates, or let's say for all eukaryotes or all bacteria, you come to what I'm calling homology and descriptive horizons. Now, strictly speaking, these two notions are not the same, and that's why I've given them different names, but they are very closely related. When you find you cannot describe something in a biologically realistic way, in effect, that's nature talking back to you. And it may be that you've discovered a fundamental discontinuity in biological form and function that gives a signal. In effect, nature says you can't actually describe what it is you think existed. If you could, you'd understand it. But the fact that you can't describe it tells you there's nothing there. Now, that descriptive horizon is very closely related to what I'm calling the homology horizon, as I'll illustrate. So these are different notions, but they're very closely related to each other. So before we get to biology, let me just give you a, I hope, intuitive example from mechanical engineering. Here is a single cylinder gas-powered, petrol-powered engine. And you can see the cylinder here. This is where combustion occurs. Combustion drives this piston down as the petrol or gas combines with oxygen. They're ignited. This is driven down. That turns the crankshaft, which will then power the flywheel and so forth. And this is familiar to anyone who studied auto mechanics or internal combustion engines. But what I want you to notice looking at this drawing from Wiki is the variety of shapes that are present. And the reason there's a variety of shapes is that the global function of turning a crankshaft via burning fuel requires a whole range of different shapes to, I mean, you can pour gasoline on your garage floor and drop a match and you will have combustion galore, but you're not going to turn a crankshaft. To perform that kind of specialized mechanical work, the system has to be constrained in various ways. And there are a variety of geometries that, whether you instantiate them with particular metal alloys doesn't particularly matter. What matters is that they are brought together in the right arrangement for that particular function. So we could explode this diagram into piston, cylinder, crankshaft itself, the spark plug, and so forth, and you will have sort of a necessary set of parts for that particular function. And we can see this actually not just in mechanical engineering, but in natural language. So let's say the meaning that I wish to convey to you is to warn you about the big dog that lives next door to me here in Chicago. And I actually do have a big dog just here to the north, the very next house over. And the meaning I wish to convey to you is don't startle him. He might bite you, okay? Something he would want to know. All natural language that we know has a core set of, I guess you could call them irreducible modules of meaning that we combine and recombine in various ways to convey sense to someone we're speaking with or writing to or so forth. And one of the challenges of describing the evolution of language historically, and this is the case today, is any functional language, as you work backwards in time, comes to a core set of I'll just use the word modules, words that cannot be reduced further, because the language at that point ceases to be a language. And you could see this with this sentence just by subtracting nouns one at a time or taking out the verbs one at a time. And you will come to a point where suddenly the meaning will be lost. Now, you might not need the adjective big, but you certainly need the noun dog. You need the information about the proximity of that animal and so forth. So I haven't done this in this thought experiment, but it should be intuitively clear to you that any functional language will have a core set of words below which it ceases to be a language. And the question is, can we describe that core set in a way that we still have the language function present? Well, we see the same thing in biology. Now, I realize that because I'm not in PowerPoint, I can't unfold this logic tree the way I wanted. So actually, that's going to be a problem, Subor, because I have a lot of animations in this talk.
Yeah, okay.
So let me try again to share my screen. Can you see my title slide?
I can't see the title slide, no. It's the same as before. Yep. Nope. I've got your...
Yeah, the problem is this is supposed to, there's too much information here all at once.
Sure.
So I'm not sure why I can't just go to my PowerPoint. It's really frustrating. If I'm sharing my screen, you should be able to see what I'm seeing.
Yep. Yep. Oh, you removed it now. There's something that I could see.
We're getting this weird infinite tunnel here.
Yeah.
I'm very sorry. I wish we should have tested this before.
No worries, no worries. Maybe if you move your cursor and you explain each point.
All right, let's just do that, and we'll do the best we can. So, how do these two horizons relate to each other? Well, here's a question that we can ask about any two sequences or any number of sequences. Do they share a common ancestor? The answer is yes. Here are sequence A and sequence B, but it really could be N, any number of sequences. We have to describe their common template. Now, either we can describe it, sequence X, or we cannot. If we can't, if unknown appears in our logic tree, we have come to the descriptive horizon. So I'll give you an example. Let's say we talk about delta hemoglobin, which is an oxygen carrier in the bloodstream of humans and chimps. Delta hemoglobin in humans and chimps differs by a single amino acid. So effectively, if I were to show you a three-dimensional diagram of delta hemoglobin from a chimp, it would look almost indistinguishable from human delta hemoglobin. Now, if I took delta hemoglobin and cytochrome C, let's say, or a bacterial protein that I'll have an example of later in the presentation, it would be very much harder to homologize those two geometries or topologies than it would two delta hemoglobins. Delta hemoglobin is a natural kind, and when you describe it in any mammal, it's going to be largely the same protein with some minor differences. So if I say I can homologize delta hemoglobin, I've got a sequence X that is the common ancestor of putatively of both. That won't be the case for cytochrome C and delta hemoglobin. So I would have arrived at unknown and I would have hit my descriptive horizon. Let's come over here to know. My question, do these sequences share a common ancestor? My answer is no. Why? The next question in the logic tree is, why are A and B not homologous? Give the reasons. They are not homologous typically. One second, please. They're not homologous, I'm sorry, because of the function they perform. When essential function appears in your logic tree, you come to the homology horizon. Now, as I said a moment ago, these two horizons are very closely related. And we can see this classically in the problem of finding the common ancestor for the animal bilaterian phyla. So here are 6 groups, bilaterally symmetrical phyla that were present in the Cambrian explosion. Here are arthropods, chordates, here's a brachiopod, and so forth. Some of these groups are extinct. This group, for instance, has no extant representatives. And they are thought to share common ancestry in something called Urbilateria. Now, this is a hypothetical construct. We don't have any fossil evidence for it. In fact, it's very hard to describe, even hypothetically, sort of in terms of, an outline on a whiteboard, because what happens as you start to give this hypothetical common ancestor features, it will tend to pull towards one or another of the known bilaterian groups. So there are 2 ways of drawing this here. This one just looks like a balloon. All it has is a through gut. It's got a mouth and a through gut going to an anus somewhere down here. This representation has paired sense organs, a primitive heart, and so forth. There are more details. But if we gave this even more details, it would tend to want to turn into a chordate or an arthropod or some of one of the known phyla, because after all, those are the creatures we know that can actually make a living in the real world. So we have again a problem of description and a problem of homology. Right here, you've got a major difficulty. This chordate has an internal skeleton. The arthropod has an exoskeleton. I would say it's effectively impossible to homologize an internal and an external major structural element. So you see the problem here with respect to whole organisms, but it comes up also with proteins. And it comes up with molecular machines, the molecular machines of the known living state. And you can see this in a very interesting discussion that occurred 16 years ago between George Church at Harvard, the geneticist Craig Venter, and the late Robert Shapiro, who was an origin of life researcher at New York University, who died, I think, in 2011. But fascinating discussion about the ribosome. So here we have 3 ribosomes broken into their large and small subunits. These are the very large RNAs that provide the skeleton, and these are very large RNAs, thousands of nucleotides long. The ribosome is effectively an RNA machine with proteins in supporting roles. So this is a bacterial ribosome, again, large and small subunit, an archaeal ribosome, another prokaryotic ribosome, and this is yeast, the ribosome in yeast, each again having a large and small subunit. The ribosome is the molecular machine that defines the living state. Viruses lack ribosomes, and they have to hijack a ribosome from some cell to replicate their genetic information. So if I had to pick a molecular machine that was all important to life on this planet, I would start without question at the ribosome. Now, the question is there a simpler form of this machine that we can describe? A. B, if we can't describe it, how are we to understand the homologies that exist between these different ribosomes? And actually, they may look grossly similar. These are actually remarkably different machines, but that's another lecture. So here they are, Church, Venter, and Shapiro. And in their conversation, Church points out, and again, this is 16 years ago, so the signal from nature is much stronger today than it was in 2007. He says, look, in every genome that we've examined so far, when we look at the genetic instructions for building a ribosome, we need these 3 very large RNAs, polynucleotides is what he calls them in the conversation, and at least 53 accessory proteins to make up this machine, the ribosome. And it seems, he says, as if we've hit a lower plateau of complexity that we cannot go beneath, where we can't or we do not find any examples of ribosomes simpler than this. So he's talking about a descriptive horizon that seems to be coming into focus. And Craig Venter says, yeah, you're right. You can't get below that. That 3 large RNAs, 53 protein threshold. But we need, for the origin of life, to have something that will accomplish replication. And Church says, well, that's a problem, because this looks like irreducible complexity. We have to find a ribosome, he says, that can do its trick with less than 53 proteins. What does Venter say at that point? At that point, he defaults to a theoretical model. He says, well, in the RNA world, you didn't need ribosomes. Now, there's actually no evidence for that, in the sense of someone has synthesized a ribosome that doesn't have proteins, that can take nucleic acid and turn it into protein. So this is strictly a hypothetical entity that Venter is calling on. And he's saying, once upon a time in the RNA world, really prior to the protein complexes that we know today from cell biology, something else was doing the job. And Church says, nobody's done that. No one has constructed a ribosome that works well without proteins. Now, that was the case in August 2007. To my knowledge, it's still the case today in May 2023, that actual functional ribosomes bump up against this lower threshold of complexity, below which the essential functions of reading messenger RNA and constructing proteins, those are lost. And then Shapiro chimes in. Shapiro was a well-known skeptic of origin of life research, even though that's what he himself did. He built a reputation as a kind of village gadfly about the work of his colleagues in the origin of life field. He said, look, it's just not going to happen. You are not going to form a ribosome spontaneously. That would be like an eye just coming together spontaneously. Now, what's the take home? A descriptive threshold has been reached. You've got a molecular machine that does a particular job for you in all cells that we know. It has certain features that you cannot do without. It may be possible to go below that threshold, but so far no one has succeeded in describing how to get below that threshold. And therefore you've bumped up against something where it looks like nature is telling you, if you want a cell at all, you're going to need a machine of this complexity and not something simpler. So that is the essential function that gives you the homology horizon. So you can see the description horizon and the homology essential function horizon are coming together at this point, which I think is very significant. All right. We can expand this away from just the ribosome to the full complement of molecular parts that are needed to run a cell. Now, in 1995, so I guess almost 30 years ago, 28 years ago, Stuart Kaufman, who does quite a bit of work on the origin of life, saw this very clearly. He was an early skeptic of the RNA world hypothesis. And he said, the leading candidate for the origin of life right now is the RNA world hypothesis. And it has evidence in its favor, such as the fact that the business end of the ribosome, the peptidyl transferase center, is actually an RNA complex. It's an RNA, a ribozyme, that is where peptide bonds are formed in protein assembly. That's a piece of evidence in favor of the RNA world hypothesis. But he says, I don't buy it. And the reason I don't buy it is rarely talked about. All living things, he said, seem to have a minimal complexity below which it is impossible to go. Now, his natural model for a minimal complexity cell, I'll show you in the next slide, but the key notion here is you're taking parts away, you're going down, 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 and you come to this descriptive and homology horizon. Descriptive, you can't say what's beyond it. It doesn't seem to be anything coming back to us from nature that allows us, without the aid of speculation, to go beyond that blue line. That's the descriptive horizon. The homology horizon is the reason we can't get beyond it, is there are essential functions necessary for the living state as we understand it that we can't do without. So again, these two horizons are coming together at this point, and they're both mapping out the same lower threshold. Okay, what's his example of a minimal cell? It's, the generic term is Pleuramona. The actual systematic term, the Linnaean genus, is Mycoplasma. So Mycoplasma is, I don't know if it's still the simplest free living cell. There may be others that are slightly smaller, but really throughout my education, Mycoplasma was taken as a model for the simplest free living thing that we know. Very stripped down cell, bacterial cell, about 580,000 base pairs of DNA. So there are viruses with more DNA than Mycoplasma. Mycoplasma, to keep it alive in a lab, the culture media are a rich buffet of biomolecules. All amino acids, for instance, have to be supplied to Mycoplasma because its natural environment is as an obligate parasite in humans, for instance, of the epithelia of our airway. So one species of Mycoplasma is Mycoplasma pneumoniae, infects the epithelia of the airway, or Mycoplasma genitalium infects the epithelia of your genital tract. Those are two places you can have a Mycoplasma infection. In that natural environment, it's bathed in nutrients. So it doesn't need to make amino acids for itself. It can scavenge them from its environment. So it's jettisoned the internal hardware, amino acid metabolic pathways, for instance, that a truly free living cell, bacterial cell, would need if it was out in my backyard here in Chicago, let's say. But you can grow the cells in culture. It's possible to do that. All right, so Kaufman is saying, let's take Mycoplasma, or Pleuramona, to give it the generic name, as our example of a simple cell. Nonetheless, even though it's very stripped down, there is this discoverable lower threshold of complexity below which we cannot go. Why is it there? Well, here's one of the reasons. This is the protein parts list for Mycoplasma. I believe it was the first species to have all of its DNA mapped by Claire Fraser, who at the time was Craig Venter's wife, at the Institute for Genomic Research in the mid-90s. I believe I'm accurate. I should check this when I send the references to Subor. The Mycoplasma genitalium was the first species, because of its relative simplicity, to have all of its DNA mapped. And they did the sensible thing. They broke down the parts list in terms of basic functional groups. So I realize you can't read this, but what this represents are proteins organized according to functional process. So this very large list of proteins here, these are all the proteins that go into making up the ribosome itself. Here's an easier way to see it. So these are the basic functional categories of any free living cell. You've got to have your boundary between yourself and the world. You've got to be able to divide. You've got to be able to convert raw materials into form that's usable to you, replicate and translate your DNA, transcribe it, and so forth. So these and other functional categories are constitutive of the living state. Now, what do the parts look like? Okay, you can go to Protein Data Bank and download the three-dimensional representations of all of those proteins on that parts list. And the thing that will strike you if you're paying attention is they're very different one from another in terms of their three-dimensional geometries or topologies. So here's a protein that is involved in the life cycle of bacteria. As you know, bacteria reproduce by fission, cell division. One key part of that process is localizing the midline, finding the 50% point along the length of the cell where you're at the middle and you're going to have a complete chromosome in one half and a complete chromosome in the other half. And then you will form a septum at your 50% point, your midline, and divide there. And this protein is involved in doing that task of localizing the midline so the septum can form along which cell fission will occur. Okay, so pay attention to that shape. You've got alpha helices here, the cylinders. Beta sheets are these little arrows here. Look at that shape and compare that to this. All right, these are not the same molecular tool because they're doing different things. So this is actually a multi-protein complex. It's a chaperone, and that's its generic name. And it associates with the ribosome during the production of proteins because many proteins need to be isolated from the cellular environment, which is very crowded, to fold properly. So the chaperone associates with the ribosome, and in this central chamber here, provides a protected environment for the protein to fold, to take up its normal folded conformation that's necessary for its function. And then once it's folded, the chaperone releases it into the cell to perform its job. So both of these are essential, right? Remember from my logic tree, when you come to the homology horizon, the reason things are different is because they're doing different things in the cell. The reason these proteins are different is because the job they're doing is different. And the same would be the case for elongation factor. It's not the same molecular tool as the other two I just showed you. Why? It's doing a different task. Now, we could, go to the protein data bank, and there are these gorgeous illustrations from the scientific artist David Goodsell that I strongly recommend to you. This is one that he did a few years ago, and you just work your way around the page here, looking at these different essential proteins. And what should be just jump off the page at you is how different they are in terms of their shape and size. The reason for that, of course, is go back to the functions they're performing. They're not doing the same thing. Ergo, they don't have the same three-dimensional form. They don't have the same size. They don't have the same partners in terms of the functions or the tasks that they will be doing. Here's a more recent diagram. Same principle applies. You've got here, these are proteins that service DNA, polymerases, and so forth. You've got some very large structural proteins here. It's remarkable, and I strongly encourage you to do this. Protein Data Bank is made, it's very accessible to anyone who wants to go poking around there. Download some of these ribbon and barrel or three-dimensional diagrams, or look at David Goodsell's work, and you will see a tremendous diversity of shape and size. And the reason for that, you go back to the functional requirements of any real cell. Lots of different tasks to be performed. They can't be performed by the same molecular entities. Just the way earlier in the talk, before the crisis with the slides, I showed you that engine, okay? Or just go in your garage. I could walk a few steps over that way and go into mine, open my toolbox, and I'll find a variety of tools there which don't share the same shape. Again, this is a very elementary point, but it's critical. Why not? They're not doing the same thing. Okay. Now, if we start with organisms, maybe it's a single E. coli, a single bacterial cell, or it might be an animal, very much more complex. There are demands made by the organismal state that point downwards towards its parts. So there are proteins in me that I can't do without. Lots of them I can, in the sense that my global set of functions doesn't require the presence of that protein. But if you work your way through your essential functions list, you find that you're going to have a list of parts that you can't do without. And that principle of can't do without, that's top down. That's the organism telling its lower level entities, I need you there for this role, to transport oxygen, to transcribe DNA, and so forth. And, the individual parts that are performing these roles are not going to be the same, because they're not doing at a sort of basic chemical level, they're not doing the same kinds of tasks. Okay. So we looked at 3 of these: this cell division protein, elongation factor, the chaperone. We didn't look at this one, but we could lay in on this slide, if we had the room, dozens of other proteins, and you would see, just from visual inspection, they're not the same. All right, now let's take this back to homology. We have a conflict within evolutionary theory between what biology tells us at the functional level and what our preferred historical geometry is telling us as we go back in time. So as we go back towards common ancestors, the governing principles say infer smaller, simpler, fewer entities. As you work your way back through evolutionary history, you are going to want to find smaller, fewer, simpler entities on back to Luca. And this will create problems for your understanding of homology. Now, if we think about a timeline of the science, molecular biology and genetics grew up entirely within a framework of universal common descent and the premise that the last universal common ancestor actually existed. So my timeline here starts with Darwin and the publication of The Origin of Species. Mendel is rediscovered around the turn of the 19th to the 20th century. Actually, his work was done back here in the mid 19th century, but really almost entirely ignored until it was rediscovered at the beginning of the 20th century. And it's arguable whether Mendel was or was not a Darwinian. I think it's an interesting historical question to explore. We can't do so today. In any case, Mendelian genetics, neo-Darwinian theory, here's Theodosius Dobzhansky, Watson and Crick, Frederick Sanger, Linus Pauling and Emil Zuckerkandl, Margaret Dayhoff, all these figures that provided the foundation for modern evolutionary genetics and comparative genomics. All of that work was done within the framework of universal common descent and Luca. Now, the reason that is significant is those deep theories. All life springs from a singularity 3.5, let's say, billion years ago, constrain what you're allowed to infer about things like homology. So a pre-Darwinian definition of homology, the form is similar because of the task that it performs. So classic example would be the pentadactyl limb. 1 bone here, 2 bones here, 5 bones here. All right. The pentadactyl limb, the homologies of the pentadactyl limb were worked out long before The Origin of Species was published. And one candidate explanation for those similarities was the functional role performed by these bones and their associated muscles and so forth. Well, that all shifts with Darwin to a primarily historical explanation. I talked about that last time. And what arises as a consequence are deep theoretical conflicts. Because let's say, and I wish I could do this as my animation, but I'll just walk you through it. I just have two here, but of course it can be any number. I've got two sequences of genes coding for proteins performing particular functions. Now, I can extend that functional claim back in time. So I start here with what I am able to observe directly, and I extend that functional claim back in time. But I run into a conflict with the theoretical demand of universal common descent, which tells me smaller entities, simpler, and fewer. Because you're going to want to work your way back to Luca and have Luca be not a complex organism with, tens of thousands of genes coding for a whole range of different proteins, but you want Luca to be as simple as it can be and still give rise as the ultimate ancestor to all life as we know it. As I extend my functional claim back in time, that's my yellow arrow here, I face a conflicting demand from universal common descent, and I get a collision right here, where universal common descent requires me to change my functional description because it's too complex for what must have existed according to universal common descent at that time. And you can see this conflict everywhere when you go into the molecular evolution literature. Now, is Luca necessary? Right? There are proposals on the table today that from evolutionary biologists, not people persuaded of intelligent design, but within the naturalistic program of explanation, there are proposals on the table today that Luca did not exist. So for Doolittle, a Canadian evolutionary biologist, who's actually a foreign member of the National Academy, quite senior figure, and one of the leaders in challenging the single tree geometry rooted in Luca, says, homology is a funny concept. It looks like we need to trace everything back to a founder genes present in the universal ancestral genome. And he says, the universal ancestor, this Luca entity, represents a horizon beyond which we can justify not looking. In other words, once we get to Luca, then we have to stop. But he says, but what if Luca didn't exist? He goes on. This is a quote in the blue box here is on the next sentence in the article by for Doolittle. If there was no ancestor, how can we avoid thinking about the possibility that all genes are ultimately derived from a single short RNA? If this is true, all genes are homologous. Now, here you see in this article from Doolittle the breakdown of the homology concept. Because what's happened is genes that are sequences that today we would say exhibit homoplasy. In other words, they don't derive from a common ancestor. Ford Doolittle is saying if we go far enough back in time, these concepts of homology and homoplasy actually collapse and everything is homologous to everything. Now that, I suggest, is not a good position for evolutionary theory to be in. But I want to show you the consequences of Ford Doolittle's view and similar views that get rid of Luca because it's hard to see how a single cell could have been ancestral to everything that we observe today. That's other talks that I've given, and maybe I can go into this in more detail in this series. But just for the sake of today, this is a figure from an article published by Doolittle, actually almost 25 years ago now, where he shows this kind of mangrove-like pattern in the early history of life. So up here are eukaryotes. It hasn't been reproduced in this cartoon, but these are eukaryotic groups here. Here are the bacteria. Here are the archaea. So we have the three major domains. And as you go back, you don't converge to a singularity. You don't come upon Luca. Instead, you have this multiple starting point geometry with a lot of interfingering where you get, for instance, during the formation of the eukaryotes, you have a bacterial cell, an alpha proteobacterium, according to standard theory, being engulfed by some host, some single-celled host that gives you mitochondria eventually, and the eukaryotic cell type goes off to form the domain eukarya. But you've got all kinds of, here it is, this branch. Bacteria gives rise to mitochondria, also to chloroplasts in the eukaryotic domain. What I want you to notice are these starting points. Notice that they don't go to a common ancestor. Here's Doolittle. I believe this is Doolittle's own drawing from his hand. Bacteria, eukarya, archaea, the three major domains. And again, this wild interfingering of different lineages. As you go back, you come to, I don't know if you can see it there. I have a slide. There should be 5 green dots at the bottom of the figure there. These are not coming together. Now, what does that tell you? Well, if we go to the next slide, here's the standard picture. You have the prebiotic milieu giving rise to various abiogenesis pathways chemically. So this is chemical determinism at this point. There's no biological descent because life doesn't exist yet. So this is being governed by chemistry. And if you remember, as I said earlier, chemistry doesn't have the same spatiotemporal. I may have said it in part 1 and not in this presentation. I'm sorry, I've been giving a lot of talks today. Biological descent as a causal relation is different in character spatiotemporally from chemical process. Chemical process is not bound to any particular space-time location. So you've got chemical pathways here, and then you have Luca, and then you have all descent. Luca, by definition, is the ultimate ancestor for all of life on Earth. What happens now if you eliminate Luca, as Doolittle wants to do? This happens. And you have multiple independent starting points. I have 6 here. No, 7, excuse me. Conceivably, you could have thousands, right? You've got an early Earth where prebiotic processes are going, they're chemical. You move your probability bead over towards 1, so you don't have to be in any special locale. Let's say you've got submarine vents, alkaline submarine vents all over the globe, churning out biomolecules, or not biomolecules yet, churning out possibly biologically relevant precursors. And you could have thousands of independent starting points. I did 7 for the sake of the slide. And these never share a common ancestor. Yet they're all producing proteins. They're all producing nucleic acids and so forth. What that tells you is as you go back in the history of life, far enough back, biological descent, as we understand it, ceases to operate. And coming bottom up from chemistry are multiple independent pathways. And they're giving rise to the same entities. Because as you raise the probability of these events, move it away from 0, right, which is where Richard Dawkins and Ernst Mayr and Jacques Monod and other neo-Darwinians had it. We're just lucky to be here. We won the cosmic lottery. Be grateful that it happened on this planet, right? But it's never going to happen again. Take your bead on your probability interval and move it away from 0 and over towards 1 in a kind of Stuart Kauffman, Christian de Duve deterministic scenario. Life wants to happen, but it's happening multiple times independently, and these lineages now are going to interact, and they will interact successfully. Okay, in this case, losing Luca, you lose homology more or less entirely. And homology and homoplasy, these concepts that we need to keep distinct to have a coherent science, they go like this. They collapse into each other and they become indistinguishable. And we end up with a very unhappy situation because again, thinking now as evolutionary biologists, our fundamental commitments in the naturalistic program of explanation are forcing us to assert contradictions. So I think that's my last slide. Oh no, this is my last slide. My little house of evolution here, which I showed previously, or I showed in the first talk, available at YouTube. I'll just focus on this one because that's what I've been talking about for the past 10 minutes or so. These two notions of causality, physical causation prior to biology and material descent after life comes into existence, they end up living very uneasily with each other as you work your way back in the evolutionary narrative. And this is something that I've talked to evolutionary biologists about. And I can tell you that when they start to think about it, it can be very unsettling.
Yeah.
Unsettling in a way that personally, as an intelligent design theorist, I don't worry about, because I don't put the burden of causation entirely on undirected process. In that sense, you can think of the intelligent design framework of possibility in a Venn diagram as the big circle. Within that is physical process as a smaller circle. Having the big circle gives you a much greater range of causal possibilities, much greater intellectual freedom, much greater facility to try out different kinds of causes with respect to the phenomena that you face. Anyway, I apologize. This is my last slide. I apologize. Next time, Subor and I will work out the tech problems that we face today. C'est la vie. We'll do better next time. But I got to my end slide, and we have a little time for conversation.
Brilliant. Thank you so much. I know you're recovering, and you had some live streams ready. So I only have one question for you, just to make things simple. And when you're better next week, we can have a longer one. You spoke about the lower plateau of complexity, and you see, this is one of those problems that, it goes back to essentially the whole problem of, if we are going to stick to a naturalistic understanding, then we're going to have to find something less than this, right? And there's theoretical problems with that. However, if we are going to have the wider circle and a wider range of tools, to understand the data, then we could actually say, well, okay, now we've reached a point where we have empirical evidence for irreducible complexity, right? So would you say this is the entry point at which we need to have the largest microphone, have the largest loudspeakers, to say, right, this is a point that we need to have a theoretical discussion, not just about the data, but about the underlying metaphysics, because it's not going to get communicated by the data alone.
Yes, you come to a point, and you can see this in other sciences, where they come to a fork in the road, if I can use that metaphor. And one of the forks says, stay with your existing metaphysics and make the best of it. The other fork says, you've got to go to a deeper level and reconsider or reevaluate the assumptions on which you've been operating.
Yeah.
And I do think that biological evidence eventually brings investigators up against or having to confront their metaphysical framework. There are many instances in the history of science where people just went down with the ship.
Yep.
Right? They are so deeply committed to their metaphysical framework, and by that I mean literally meta, above the physics. It's the point at which you move into the philosophical framework, into the assumptions about what counts as an explanation, what could be a possible cause. And these have scientific content, of course, but they're not wholly scientific. I'm sorry, I'm using a range of different metaphors. One way to think about this is with respect to playing a sport. For those in the UK, football, soccer for us, cricket. I don't know how many rule changes have been instituted in the game of cricket over its history, but I can tell you in baseball, here in the United States, just this year, there's now a rule that the pitcher has to throw the ball within, I think it's 30 seconds, or maybe even shorter than that. I don't know the rule exactly, but the problem was pitchers would stand on the pitcher's mound holding the ball and just wander around and look over at first base and scratch their head. And the games took forever. So top down, the professional game of baseball said, you've got to throw the ball within this well-defined interval. So rule changes happen all the time in sports to improve the game. It may be that rule changes, such as methodological naturalism being suspended, may be necessary to let biology follow the signal of the evidence, such as what Subor, you said, the reality of irreducible complexity. And one of the things I love about intelligent design, even though we get beat up and insulted and not literally beat up, God forbid, but you know what I'm talking about. That we can come to this point of contact between the philosophy and the science, and there are so many interesting questions right at that boundary line.
Absolutely. That's a fantastic way of giving an answer to a question which I think we could give an entire talk on, the fork in the road, the metaphysical fork in the road and the implications of it. But I'm not going to hold you any longer. I know you're recovering. You had a tough week. So thank you so much for your time. It's a delight to have you as always, Paul.
Thanks.
Until next time, Assalamu alaikum wa rahmatullahi.